Hello, everyone. Thank you all so much for joining us. My name is Julia Chen. I am the Activities Director at the Yale Club, and I'm so excited to welcome you all to our talk today. Today, we are excited to welcome Dr. Paula Kavathas. She is a professor of laboratory medicine, immunobiology, and molecular, cellular, and developmental biology at Yale. She received her PhD in genetics from the University of Wisconsin-Madison and did a postdoctoral fellowship at Stanford University before joining Yale faculty in 1986. Her research is on immunotherapy in lung cancer. She is the co-author of a textbook entitled Immunoepidemiology, board member for the Yale Poorvu Center of Teaching and Learning, and on the executive board of SWIM, Status of Women in Medicine. 10 years ago, Professor Paula Kavathas started a course entitled Immunity and Contagion at Yale to educate the non-science major on the immune system and microbes they encounter either as pathogens or as partners, the microbiome. A recent article on the course in Yale News titled Classroom Lessons on Contagion couldn't have come more timely, highlighted educating students about pandemics and their historical context. And today she's joining us to speak on the course, her philosophy, and what education is necessary to survive a pandemic. And with that, I'll turn it over to you, Professor.
Thank you very much, Julia. And thank you for inviting me. And thank you for everybody who's on the call tonight. I am a faculty member over at the School of Medicine. And so our expectation there is to do research and teach graduate students. So how did I get involved in an undergraduate course? Well, in 2006, one of my daughters graduated from Yale College, and I got to know a lot of her friends, and I was inspired to do a course. I've always been interested in science literacy. For 18 years, I started and ran an outreach program into the New Haven public middle schools, where graduate students and postdocs would go to the school and do some projects on genetics. And so I decided to teach a course. There was an immunology course that was really for science majors, and there were microbiology classes, but there wasn't a course like I was going to design. And I decided to combine immunology with microbiology because our immune system evolved to deal with microbes that would invade us and make us sick. And so we needed to know both about the immune system as well as about different microbes and pathogens. So I started that course, and I focused also on microbes that cause pandemics. And I'm going to show I have a number of slides to show, and hopefully we'll have some discussion as well. I thought I would just show this one slide of my Zoom class. As you know, Yale didn't return from spring break. The students didn't return, and so we were given 10 days to go online and teach. And this is a subset of the 45 students that I had in my class. And it was actually fun, a challenge to be teaching online. And I think online learning now is going to be much more prevalent, either as classes by themselves or as hybrid classes. Okay. So today I thought I would talk a little bit about the course and some of the topics I covered that are relevant to pandemics, and then address the question of why does science literacy matter during a pandemic? And I'm hoping we can make a little bit of a discussion on that. And also, does Yale's education prepare the graduates in terms of science literacy? And that's another thing I thought we could potentially discuss. So I wanted to just start with a poll. So 1, excellent. That's 5%, 18% good, and 33 fair, and 55 poor. So I think it's a small sample size, but I think it's indicative of the fact that we're not doing a good enough job of preparing our college graduates to be scientifically literate. And I think we realize more and more some of the challenges that we have today where science literacy does matter. I'm going to talk a little bit about the course, and then we're going to talk a little bit about science literacy and pandemics and what we could do better at Yale. These are the topics that are covered in the course. The first is how the immune system works. And then we talk about specific microbes and pandemics. And we do HIV/AIDS. We do influenza in the 1918 pandemic, human papillomavirus and cancer vaccines for human papillomavirus. Human papillomavirus, there are high-risk strains that are found in head and neck cancers, in cervical cancer of women, and really fantastic progress for making a vaccine to protect people, which is a real advance. And then we have the coronaviruses and COVID-19. And I really wasn't teaching this before, but in the middle of the semester, I realized I better start teaching this. And so I invited the head of the virology lab at the Yale School of Medicine to come to the class, and she talked about coronaviruses, the ones that cause colds and the ones that are much more lethal, SARS, MERS, and now SARS-CoV-2, which is the name of the virus that causes COVID-19 disease. And I also talked to the students about immunity to this virus and also different tests, how the tests work, what's an antibody test, what's a test for the virus, so that they would be educated on things that they were reading also. And then I also talk about issues related to the immune system, such as immune disorders, autoimmunity, autoinflammation, hypersensitivity, which includes allergies and asthma, immunodeficiency diseases. And then about cancer immunotherapy, which is a very exciting area of immunotherapy now for cancer patients. And then I also talk about the microbiome. We live with trillions of microbes within our GI tract, on our body, in our mouth, our scalp, many parts of our body. And we live harmoniously with these. And so we can't just view microbes as something to fear. Certain microbes, yes, we should fear, but others we live in harmony with, and we have a symbiotic relationship with them. Okay. So just to, I'm gonna, as I said, talk about some of the pandemics, cover some things for about 20 minutes, and then we'll move into discussion of literacy. So our immune system has two components. One is the innate, it's called, and this is in all species, including plants. And it responds within hours. There's certain cells and certain ways of detecting classes of microbes. And we actually have receptors for detecting these microbes, just like you have receptors for taste, receptors for smell. We have receptors in our genes that we have genes in our genome that create these receptors to detect microbes. And then invertebrates, which are about 1% of all species, the adaptive immune response developed. And this takes longer, 5 to 10 days, but it's very specific, and it has specific ways of fighting the microbes. And we also develop memory. And that's really the basic for vaccination, because these cells develop into memory cells. Sort of like our nervous system, we can develop memory to things. Well, our immune system also can develop memory. And this time when we get reinfected, the response is much stronger and much quicker. And so those are the two basic arms of the immune system. And I just want to define receptor. So the idea is we have a structure even on the surface of our cells or inside our cells that binds something. In the case of the receptors for microbes, maybe they bind a bacteria or they bind some component of a microbe. And then they send a signal into the nucleus where the genes are located, and then the cell makes a response and you get a response. So that's how we detect microbes. So to start with the AIDS epidemic, I was a postdoctoral fellow at Stanford from 1980 to 1986. And this was the beginning of the AIDS epidemic. And what people were finding was that there were healthy young adults who were getting diseases that were rare and rarely found in young adults. And so they knew something was wrong. And they were trying to figure out what was happening. And so they knew it was an infectious disease. They figured this out. And so what do you want to know when you have a new disease? One, you want to know what's causing it and how is it transmitted between people. And this is something that we are also asking today. Where did it come from? How does it affect the immune system? Can we develop a diagnostic test and a therapy or vaccine? And then epidemiology is studying how it's spreading in the world and how we can stop the spread of a disease. Fortunately for HIV/AIDS, they don't have a vaccine, but they did develop antiretroviral drugs. These are drugs that specifically target proteins that the virus makes. And so they bind to those proteins and block their function, and the virus can't multiply and continue to infect. And so the heart, which is the highly active, has at least 3 or 4 different antiviral drugs. And this has made a huge difference in our fight against AIDS. And so this is just showing a death curve for for individuals in the US. And you can see this is how during the early 1987, you started seeing an increase in the death rate because of AIDS. But then with antiretroviral therapy, it really dramatically went down. And this is comparing it to other things, other kinds of diseases such as injury and cancer, etc. But it's still a global problem. We still have 37.9 million adults, at least in 2018, that are infected. Now, one of the characteristics of this pandemic is that there was a lot of stigma, despair, denial. It was thought it was only one group of people, and that was later shown to that this virus spreads to anybody. And so we've had to deal with discrimination and stigma in this disease and try and illustrate how negative that is to combating a disease. And I take the students to the Yale Art Gallery to see art in relationship to AIDS. And so one of the pictures that we look at, the students look at, is this of Cookie Mueller. And if you look at her, she looks like she's a well-dressed, woman. And turns out that she died from AIDS. And this was her friend, Nan Golden, who took this picture of her and created this imagery. And so I try and make the pandemic real for the students. They've never experienced a pandemic normally, but this generation of college students is now, and they're getting a sense of what it is to be in a pandemic. Another pandemic I talk about is the 1918 flu pandemic. This is a graph of mortality and year. And what you can see from infectious diseases over time, there's a huge spike at this time. And that was because of this 1918 flu that caused 50 million deaths worldwide. In India, 6% of the population died. And it was compounded because of war. At that time, World War I was going on. People were traveling, and there were strains on people being in war. And that was a contributor. And so that's something that we talk about. And I actually asked the students to compare this pandemic versus the one we have today. One of the, this is just showing nurses from the book, The Great Influenza, wearing masks. And basically people were wearing masks. And at that time, they didn't even know what a virus was. They just knew that there was an infectious agent out there. And so they were just covering their faces with these masks. And you can imagine how panicked they were when you don't even know what's a causative agent, and they don't have the information that we have now, which is, much, much better. And the other thing about this pandemic, which was a little different, is that this is a death curve from infectious agents that cause disease. And in the normal death curve, this is the age and this is the deaths. Normally, the very, very young and the elderly are more susceptible to this normally. In the case of the 1918, it had a W-shaped curve. And it turns out that young adults were very susceptible to dying from this. And young children were more protected. And then there was a group here. These were middle-aged, kind of 50 to 60. These were people who actually had been exposed to a flu in the late 1800s. And there was enough similarity that they had some memory and some immunity that they could help them fight this infection, whereas these younger people did not. And this virus had strategies to evade the immune system. And so these young adults were highly susceptible to this virus. And this is the influenza virus, and it has an outer shell, and it has an RNA as its genetic material. And on the outside are these two proteins, neuraminidase and hemagglutinin. And N, you know how you say H1N1 or H2N3? That has to do with the type of these two proteins. And this virus mutates every year, which is why we have to get new vaccines every year. But in the case of a pandemic, it changes radically. And so this is a little schematic of the surface protein that's hemagglutinin. And the red balls show the differences in the different years of how the hemagglutinin looks different in terms of its amino acid sequence. And what you can see in swine flu, all these red balls indicated that it was very different. And therefore, we didn't have, many people did not have prior immunity that they can call on. And this led to this pandemic. And the pandemics are when these viruses have a very radical change. And there's genetic reasons for that, which I go into with my students. Now, the coronavirus also has RNA as its genetic material inside. And then it has on the outside certain proteins. And one here is called the spike protein. And this is important for infecting cells. So I'm going to talk a little bit about coronavirus, just a few slides, but I want to go back to the innate immune response because I'm going to talk about that. It's relevant to stuff that you hear in the press. So the innate immune response, as I said, occurs within hours. It sees classes of microbes, and it has specific receptors that detect those microbes, and then it makes responses. And some of the responses are to secrete what are called cytokines. And I'm going to explain that next. Sometimes the cells will lead to death. You want to kill the cells that are infected. So, hey, I'm going to sacrifice myself and die. So that infections can lead to cell death also. So what are cytokines? So just like you think about hormones that are secreted by certain cells and then they affect other cells, cytokines are like that. They're secreted by one type of cell, and then they're secreted, and another cell that has a receptor will then make a response. And there's hundreds of these cytokines. So they're the chemical communicators between cells of the body. And one class are called interferons. And because what interferons do is they interfere with every step of the virus life cycle. And the other type of cytokines that you've probably heard are called inflammatory cytokines. These lead to inflammation. And normally inflammation is good. We want inflammation because it mobilizes the body to fight a microbe. But if it's excessive, it can lead to damage. And that can be a problem. So in the case of the coronaviruses, they infect epithelial cells of our upper and lower respiratory tract. And when the virus goes in, the cells sense it and they will secrete cytokines. So how does that happen? Well, the spike protein binds to a protein on our epithelial cells called ACE2. And once it binds, it internalizes. Now we have a receptor inside one of these innate receptors that detects the RNA of this virus. And it will then lead to a response so that interferons and inflammatory cytokines are made. And this is important for fighting the response. And so this is part of our innate immune system to have this. Oops. So what other response do we make? Well, I said there's an adaptive immune response, and that is in vertebrates. And there's two arms of that. There's the cellular, which involves a cell type called a T cell, because it develops from the bone marrow into our thymus, which is an organ just above our heart. And the other is the B cell, which is involved in making antibodies. So, oh, sorry, I'm going the wrong way. Okay, so the antibodies, there are different types of antibodies. And to be protective, we want to have an antibody that's called neutralizing antibody. So in this case, the antibody binds to the spike protein and then can prevent it from binding to ACE2 and infecting cells. So that's neutralizing antibodies help protect us from viruses. And many of the vaccines against viruses are designed to create neutralizing antibodies. And that will be something that they'll be working on for COVID-19 to try and develop a vaccine for to induce neutralizing antibodies and enough of them that will also last to protect us. But the other, oops, keep going the wrong way, sorry. The other immune response are killer T cells. And we have these fantastic cells that can detect one cell that's not infected versus a cell that's infected. And they can recognize cells that are infected and they can kill them. And it takes time. So initially a virus comes in and it starts growing, and then we start slowly getting a T cell response, and that starts coming up and it controls our virus, hopefully, in many cases. And then it starts going down. We don't need the cells anymore, but then we have long-lived memory cells. And so that's the other kind of response that we need against viruses. So we have our innate responses, the interferons, the inflammatory cytokines, we have the adaptive antibodies, and we have the adaptive killer T cells. Okay, so how does science literacy help during a pandemic? I put some things up here, but I think it would be good if people want to add something into the chat as well. So one is, I think that pandemics happen, they can last for more than a year, but they do end. And I think that's always a positive, something to be hopeful. It's important to be prepared because they happen throughout history. Also, we know that different microbes have different characteristics and they can impact the immune system in different ways. And so some people have said, oh, is COVID-19 like the flu? No, it is a respiratory virus, but it's not the same. Also, I think it's important for people to understand vaccines and herd immunity. We need to understand vaccines so that people will get vaccinated. It's had a huge impact, and one of the greatest triumphs of medicine was the development of the smallpox vaccine. Smallpox was eradicated in 1977. It was a horrible, horrible disease. And I think something that's really important, and especially I think for people at Yale as well, is that science leaders need to be scientifically literate so that they can make better decisions for the people that they're serving. I think this is so, so important. I'm going to stop my screen. I only have 2 more slides. Let me do this, and then we can get involved in the chat and have some discussion. Oh, I know what I wanted to say also. Some of my students wrote quotes for the Yale News article rather. And what another thing, the value was quelling fears. So by having information. So one student wrote, "The course has helped calm some of my worries, the worries of my immigrant parents who are more prone to misinformation and have often reacted with irrational fear with regards to the virus." Another student wrote, "With the knowledge of this course, I've been able to help my family and myself understand what is fact and fiction and what is or isn't a reasonable course of action." Sorry. Another student wrote, "Good science takes time. In addition to confirming the importance of vaccinations, the most important course takeaway is that good science takes time." Another student wrote, "It gives me a lot of insight into the scientific process with regards to producing a vaccine, which is insanely relevant today. We read the book Polio, An American Story." This is one of the books I have the students read because I want them to be aware of history as well. And he wrote, "Before COVID, we read the book Polio before COVID-19 had altered our lives, really. So I thought it was crazy to read that entire towns were shutting down because of polio, but that is what is happening now." Okay, and then my last slide is I would like to sort of have a discussion. Does science education for a Yale graduate adequately prepare them? I think the poll says no. And how can we improve science literacy for our Yale students? And I have some ideas, and I thought maybe we can have a conversation on that.
Great, and thank you so much, Professor. So, I mean, if anyone has any questions or comments, like if you want to suggest your ideas for how we can improve science literacy, please feel free to put them in the chat, or if you use the raise hand function by clicking participants, I'll unmute you and you can ask your question aloud. But as you all think of your suggestions and questions, we already have a question from Richard asking if you could please discuss antibody testing and which ones currently available do you think are the best?
Okay, so there are a whole variety of tests out there now. And I know at Yale, they are trying to validate the different tests. And so I really can't specifically comment because I'm not really involved in that effort. But antibody testing is, one way they do it is they take, there's a couple ways you can do it, but the simplest is they take, let's say, the spike protein, and they have an assay then to ask, if we take a person's serum from their blood, and we put together the spike protein and your serum, and then we wash, and then we have a way of detecting antibodies. We ask, are there antibodies in your serum that bound to the spike protein? So that's one way to do it. Another kind of assay that's more complicated, but is also an assay, is to ask if we create cells that can be infected by the coronavirus. We can take the cells and the virus and put them together, and they will infect these cells. But we can first take the virus and mix it together with your serum. And if they're neutralizing antibodies, it'll bind to the virus. And now we ask, can the virus infect cells? And we can look for blocking. So we can actually develop an assay to detect blocking. I think most of the assays are just detecting the presence of these antibodies. But the question is also, are they neutralizing antibodies or not? That's also important. So there's a lot of work going on right now. And a lot of people say, what are you going to do with the information? Because we don't know what level of antibody is needed to be protective. And that's another question. And so it doesn't mean because if you have an antibody test that's positive, that you are necessarily protected. We don't know that yet. And also the validity of these tests, I think, are still actively being investigated.
Great. So I think I'm going to unmute Malik to ask a question or make a comment.
Sure. Can you hear me?
Yes.
Yes.
Hi. Thank you for taking my question. So I'm one of the non-science majors in the audience, and to me, good literacy in science is just as important as that level of literacy affecting behavioral change. So the way I understand public health is if there is a big public health event like the pandemic that we're living through now, you also have to have, behavioral change accompanying, scientific advances to combat that event. So in your classes, I just wanted to understand if you had any discussions on the sociological dimensions of, how that can actually augment and how that can augment scientific literacy and, achieve the end outcome of improving public health.
Sure. I mean, that's one reason. One is we read about polio: An American Story, because it's really discussing how people were being impacted by this disease, how people were responding. Yes, we do talk about the social issues, and that disease cannot be seen alone. It has to be seen in a cultural setting. And so that's an important dimension of science literacy. Also, I want to bring up this issue. I've been trying to think about how we can improve things, because right now Yale undergrads who are not science majors just take 2 semesters of science. It can be anything. So one of the things I've thought about, should we have a 1-year science biology curriculum for the non-science majors? They have one for the science majors, which has 4 modules, half a semester each. And I was thinking, and I've asked my students, would you be interested in taking something like this? So that it would be geared to what do we need for science literacy in biology, genetics, different, what I'm teaching, ecology, evolution, different things that we would have. Another idea is there's a major called ethics, economics, and EP&E, politics. Ethics, politics, and economics that students take. Should there be a potential curriculum that would include science and that maybe would have humanities and arts, economics and commerce, science, and then another option for students to take other courses that they would want. But we would have a kind of similar to ethics, politics, and economics, but add the word science in there, so that would be something for the non-science major. So I don't know how people think about some of those ideas, or what else could we do?
Yeah, so I'll actually read aloud verbatim something that Andrew had put in the chat. So, accepting the premise that scientific education is important, particularly in this area, making this a full semester course ensures that it will only reach a small portion of students. Why not change the curriculum to treat courses in immunology and many other important areas of study as smaller modules in which every student needs to be fluent, just as every student has to pass a swim test, which saves their lives, but they don't need to spend an entire semester. And he says that we learned a lot in just these 20 minutes.
Yeah, I think that would require a radical change in the curriculum to have small modules. I mean, I think we could still do a semester course, but with modules. And I think that, as I said, there's 2 semesters required. So if you have a full year of science with 2 semesters, you could do 2 or 3 modules for each of those. And we don't have anything like that. There is called topics in biology. But to me, I think it's important to have a certain depth of understanding, actually. And, they touch every week on different topics. But I do think that we need to go in depth to certain topics to really understand them. And so I think, but I agree that modules is a good way to go.
And then perhaps approaching it from a different perspective, we have a comment from Lisa saying that the New York State health curriculum, every student must take this course, requires the study of immune response, particularly in relation to AIDS. Would you recommend changes in high school curricula as well as college curricula? Health course instructors must have special certification, so it's not a lay person teaching.
Yeah. There is an issue of our science curriculum. They have been trying to improve it. Most students had hardly anything on immunology. I mean, really, it was just terrible. Now for AP biology, they do have some immunology on there, but that is for students in AP classes, and that's not for your, regular citizen. So there is an attempt to increase this. I think people have been scared off of the immune system because it seems very complicated. And there's certain fundamental things that people don't know. And so we're trying to, see if we can also help in that area. And I know the American Association of Immunologists is trying to develop basic concepts that we feel people should know, but still, again, that emphasis towards the science major. And I think we need to do more for the non-science major. And I think this pandemic has really highlighted how important it is for leaders to be scientifically literate.
So the question is, if someone recovers from COVID-19, can we be confident that T-cell immunity will prevent reinfection?
It's all being studied. It's being studied, but we don't know. I mean, we've only really been studying this virus for a couple of months. There was a big shift in many universities where people are starting to, but I think as the other student said, it takes time, but people are definitely looking.
And then a question from Richard: how do you test the effectiveness of a vaccine if it is not ethical to intentionally expose a subject to coronavirus?
Basically, you want to test it in an area where the virus is spreading. So, SARS in 2003, which is a coronavirus, that's SARS-CoV-1, they call it, that spread rapidly, but people who got it generally got very ill. And so you could easily identify who the people were, isolate them, and quarantine them. And so we were able to stop that disease before the vaccine was really developed, because people started working on it, but then there were no patients to test because it was contained. Now, in this case, because we have asymptomatic carriers and it's global, I think we will be able to test this vaccine just by immunizing people in certain regions. And you need to have in a vaccine, you need it's usually what's called a double-blind study, where the person giving the injection doesn't know whether or not it's the real vaccine or a placebo. That means something that isn't going to protect you. And so to really do an adequate study, you need to have both controls, people who don't get the vaccine and people that do. And at first level of a vaccine is safety, and then small scale efficacy, large scale efficacy, and then if it's shown efficacious. But I think in this case, we will have subjects to test.
And then going a little bit back to the science literacy part, I think one of the reasons that I was very excited about inviting you to come talk to us about all this was that for many people, even if they were science majors, if they're not in science now, it's probably been many years since college and they're forgetting. So how do you think we could, I guess, maybe even re-educate people later in their lives on these very important topics?
Right. I think online courses are great. And I think there'll be more online courses. I mean, I have a friend who's now said to me, you should really give an online course on this topic. And I think that that's another way we can do this, is by having online courses. Now, you hope that students will, pick up the science section of the New York Times and read some of the articles to kind of help keep up to date. But you're right, things really change. And I think the online courses, short courses where people don't have to pay, but I think for Yale, they are offering some courses for free. And I think, even if it's just to the Yale community, I think it would be good to have online courses for topics that are very relevant at the moment.
So while everyone thinks about if they have any other questions or comments, I wonder, Professor, if you could tell us a little bit about your backstory and how you got into science and immunobiology.
Oh, yeah, sure. It's all serendipity. After I graduated college, I decided to work in a lab. I wasn't exactly sure what I wanted to do, and I had a friend who was in an immunology lab, and she said, oh, why don't you come and work here? And so I interviewed and I got a job there, and I worked there for a couple of years and then decided to go on to graduate school. And I've just continued in immunology ever since. It was not even a separate field at that time. So my graduate work was in genetics. There really weren't immunology departments. And I was studying genes that are important for immune response. And so that's what I did it for my work. Now, of course, there are many immunology departments, and Yale, at the time when I came here as an assistant professor, there was no immunology department. Immunologists were part of the pathology department, and there were some in public health, but there's no department. And since I've been there, they developed a section of immunobiology and then a department. And so now we have a department. And I think it is so important and so relevant. I mean, part of the reason I'm teaching this course too, I have all the students in the beginning of the semester stand up. And then I say, sit down if you have an allergy or asthma. Sit down if you or someone close have an autoimmune disease. Sit down if you've had the flu or or some type of infectious disease, and there's barely anybody standing after this. So it's very relevant to our health, just personally, our personal health, as well as our collective health. And it affects plants. I mean, plants have an immune system. They have problems with fungi and even bacteria have viruses that infect them. So it's such an important field.
And we have a question from Debbie Rosenberg-Bush asking, at this moment, the field of public health is having greater visibility than it has for decades. Is Yale's public health school seeing an increase in applications? And is Yale Career Services Office prepared to serve and direct undergraduate students who may find the field appealing? In her day, it was not mentioned at all.
Yeah. I know that there is a global health program for undergraduates that they can participate in. So there is a program for undergraduates where they can participate in global health. I don't know about applications. I do know that there's a colleague of mine in public health who taught immunology for those students who were in public health with a focus on infectious disease. And she's emeritus right now, and someone else took over the course and they're retiring. And so there was a big discussion about whether or not they're gonna continue with the course. And there were some people, and this was last year, some people saying, oh no, we don't really need immunology. And she was saying, no, we need immunology. And so she prevailed, and she is actually coming out of retirement to teach this course in the fall because she feels it's so important for students of public health. Her name is Dr. Nancy Ruddle. She's a great scientist. She's been a leader in the field of cytokines and autoimmunity. Anyway, so she's teaching it this semester. And, she's 80. And she is now taking classes on teaching online in case we have to be online. Oh. And she and I actually want to work on a book that is similar to this course, basically Immunity and Contagion, because there's no textbook out there like this. And the first part would be immunology, and the second part would be different microbes, different pandemics, and a theme for each pandemic. And our theme for HIV/AIDS was stigma. And so I just want to throw it out there. We don't have a lot of people, but if anybody's involved in book publishing or has any connections, we just made our outline and our letter and things, and we're pulling it together, and we want to then try and find a publisher. Even if it's an ebook, I think it's relevant, and people will sometimes create courses if the material's out there.
Yeah. And I know I've heard from a lot of the authors that I work with that Yale University Press is actually fantastic and great to work with. So that's one direction to look into.
One option, absolutely.
Great. So I think that's all the questions that we have for now. So Professor, did you have any closing remarks for the group?
I'm just so happy to have the opportunity to talk to you all. And if anybody has any questions or anything that they would like to email me, I'm paula.kavathas@yale.edu. And I'd be happy to help in any way. And so I just want to say, wear masks, stay safe, and we will get through this. I'm optimistic about a vaccine. I really am. I know some of the things people are working on, as well as even drugs. I'm very optimistic, but it's going to take time, and we just have to hang in there.
Thank you again, Professor, and thank you everyone for attending. This is such important information. Share it with your friends, share it with your family, make sure that everyone is aware of this crucial science literacy information. And also join us for many of our upcoming events. They're all on the online calendar, and we hope to see you at one of them again soon. Until then, stay safe and stay connected, and we'll see you next time.